tests and see whether or not you're able to get the terminology. And these are the terms that you'd have to be really very comfortable with for the exam coming up. Okay, so it's just a matter of just reinforcing this. Fortunately, you'll get these back tonight, 10 points, lecture, so it goes on to the lab side. All right, but I think that what it does is to kind of keep you on task. And then we'll be having another group assignment coming up so that you have some opportunity to gain some additional points on that one there as well. Okay, so it's only this week and next week, and then we're done. Okay. All right. And we've got tonight, if you looked at tonight's lab on the brain, you probably noticed that there's a lot of information that needs to be learned for this next section. So, again, it's a matter of kind of compartmentalizing, learning the terminology, and then checking in that period of time how much have you learned, basically, is what it's going to be. Okay, so let's go ahead and get started. You guys have the handout. I'm going to go with the brain. Let me see what it is. Okay, and I'm going to be going through the next section on the PowerPoint. It goes very much in sequence. What PowerPoint is this? It's right in the order. It's the second one on the brain. Pardon?
Oh, I don't think it's up yet. No, it's not up yet.
Oh, okay. I can't open it up on here because I can't figure out this particular computer. In the lab, I'll go ahead and open it. Nobody, I didn't even look. I know that it's on mine. Okay, so we'll go ahead and just go with it right now. Okay, so we're going to be doing the next handout, which is on the brain. So I guess you'll have to look at this one here. I'm sorry you don't have that up for you already. Apologize for that. Okay, so we're going to start on the information pertaining to the brain. Then we're going to follow it with the spinal cord, and then the protection surrounding the brain. Okay, so that's my next sequence. So today is going to be predominantly the external features of the brain itself, and then we will on Thursday go through the mid-sagittal section and identify structures within there. Within that, you'll be looking at models tonight, and the diagrams that you see in lab, I think it's 20, are structures that you need to be able to identify on those models as well. Okay. Okay. So the brain, the cerebrum has, well, the brain itself has 4 different regions. The largest part of the brain takes up about 80% is going to be the cerebrum. Okay. We have 2 halves that are connected, okay, by way of fibers. So they connect the 2 halves so that they can coordinate activity between the 2 cerebral hemispheres. Okay. If I look at the next part, it's called the diencephalon. Got some diagrams that are going to illustrate it much better in a moment. So the diencephalon has a variety of different components: the epithalamus, the thalamus, and the hypothalamus. Now you know one structure that's attached onto the hypothalamus, and that would be the hypophysis or pituitary. So that's pretty much in the center of the brain. The brain stem is the midbrain pons and the medulla, and then the cerebellum. Okay, so this is what it's going to look like. Okay, so you can see on here. All right, so you can see the cerebral hemisphere takes up the greatest portion of the brain itself. Then you have the center region right here, which is the diencephalon, and that's when you do a mid-sagittal cut. That's when you can actually see some of the structures associated with the diencephalon. And if you're looking at parts of that, this would be the hypothalamus here. The epithalamus is over here, and then this is that pituitary that I was talking about. Where is Okay, so it's in the skeletal sabbat, okay? And it fits right into that little saddle. And then the anterior clinoid processes and the posterior clinoid processes serve as attachment points of a meninge that kind of holds the pituitary in the pocket. Okay. All right, so that would then be my diencephalon. The cerebellum is this portion here. And then we have the midbrain, and then we have the pons and the medulla, which then once it passes through the foramen magnum, becomes then the spinal cord. So those are my major regions that are associated with the brain. Okay, this is what it's going to look like in a preserved state. Now, if I were to take a brain out of a newly deceased individual, the brain actually is suspended along the internal wall of the cranium and is held in position. It's kind of like, it's got the consistency of kind of a hardened jello. So if I take it out and put it onto a dissecting pan, it would then compress by about 1/3. So it's suspended to give it its shape. Now this has already been once removed. It has been put into a formalin solution, which then makes it into this very, it looks very stiff, okay, fully enlarged portions of the brain. So you can see that there are a number of this identified on here. You'll see this on the slide. This will then be your cerebrum. You can see then the cerebellum. Cerebellum just refers to a small cerebrum, okay, so it's a small portion. The diencephalon is this little area right here, right in the middle, and you can't really see it unless you make that mid-sagittal cut. And then you've got various components of the brainstem, which then is the midbrain, the pons, and the medulla oblongata. So this then is how the brain is organized. And you'll notice that on the brain, there are a lot of convolutions, hills and valleys, okay? And these hills and valleys are very important because they serve as centers for interpretation or for motor responses. So all of the brain has been compartmentalized into a variety of motor and sensory functions. We're going to talk a little bit about how we can determine that. Okay. All right, now within the brain, I have these ventricles. The ventricles are fluid-filled spaces, okay? And the fluid that's in that space is called cerebrospinal fluid. So what that CSF does, and it's got the same composition as the plasma portion of the blood. We're going to find out what little small capillary in each one of those ventricles is going to actually produce the CSF. But when that fluid goes into that space, okay, and that space then is going to serve as a fluid, is going to serve as a shock absorber. So if I were to have trauma on one side of my head, because of the consistency of the brain being kind of a hardened jello, it will then kind of have a rippling effect, and I don't want it to damage the other half. So the water in there serves as kind of a shock absorber, okay? And it then is going to protect the brain internally. Now you see on here the ventricles have the CSF, and they're all interconnected, okay, these ventricles. And around the cerebrum and the entire brain itself is also a little layer of CSF. So it's all over, it's on the superficial to the brain, within the brain, and it then goes around the spinal cord and down the center of the spinal cord. Okay, so the function of the CSF is primarily for protection in terms of being reducing any kind of trauma being propagated through that brain. So you've got then two C-shaped ventricles. These are my lateral ventricles. They go into my two cerebral hemispheres. Okay? So then those two are interconnected by this interventricular foramen that then leads the CSF into the third ventricle. So my third ventricle is found in the diencephalon when I put the two halves together. I form this little virtual space. It's not virtual, it's a real space, okay? That then is going to be my third ventricle. So the walls of my diencephalon are going to enclose that third ventricle. Then the fluid passes from the third ventricle down the cerebral aqueduct into the fourth ventricle. In the fourth ventricle, then I'm going to have openings that are laterally arranged, and these openings are the lateral aperture that then takes the CSF and brings it around the brain. Okay? And then coming down from the fourth ventricle, I'm going to have a central canal that goes down the center of my spinal cord. So the expectation is that you follow the flow of CSF. Okay? Two lateral ventricles through the interventricular foramen to the third ventricle. From the third ventricle, it's going to pass through the cerebral aqueduct to the fourth ventricle. At the lateral apertures, down the central canal. And then it's going to surround the spinal cord, and it's going to go around the cerebrum. Now, these are spaces, and so there is a specific kind of a cell that is associated with the lining of those spaces. What cells would those be? So the ependymal cells, just like we said, epithelial cells that always line a cavity, these are ciliated, and that cilia helps to produce a circulation of the CSF within those spaces. Okay? There's a specialized in each one of these ventricles, 4 ventricles. Each one has its own special structure called a choroid plexus. And that choroid plexus is a capillary. And so those ependymal cells that are in that capillary or close to the capillary will then produce the CSF. So this is what a normal ventricular system would look like within the cerebrum. And it's actually within the brain, let's just say it that way, because it's also part of the diencephalon, it's in my midbrain, etc. Okay, so this is what it would look like in a variety of sections. Okay, so you can see here, this is a horizontal cut. So the horizontal cut going this way, remember it was a C shape that looked kind of like that? Okay, they go like that. So then this would be the anterior portion, and this would be the posterior portion of that C-shaped opening. This then would be my third ventricle, my lateral ventricles, third ventricle, and then if you go down here, we'll go down into the fourth ventricle. So this is where we would find the ventricular system. And in lab, I think it's either tonight or on Thursday night, you'll need to go through that circulation of CSF. Okay, from which does it start? And we're hoping that along that pathway, all of the openings are open, that they're patent, because you want to make certain that the fluid does move. If it gets plugged, then we start to limit the circulation, and it then accumulates in the ventricle, and it actually can press against the brain tissue itself. So in a child, while in utero, while the brain is developing, if there is a plug, if there's a closed opening in that ventricular system, then that child's brain will be compressed and you get hydrocephaly. Okay, the head becomes very enlarged. But you can check for this prior to birth, and you can actually tap in and remove the fluid if you do your ultrasounds. So the ultrasound is really important because it then is able to monitor the normal growth rate while in utero. Now, oftentimes we think about hydrocephaly associated with a fetus, basically, but it can also happen to an adult, so long as those sutures and fontanelles are not, well, let's just say not fontanelle, the sutures are not ossified. So if we don't have ossification yet, and that happens in the early 30s, you actually can have an accumulation of CSF in the ventricular system, and then it can compress onto the brain. And then one of the symptoms would be then obviously pretty severe headaches, okay, depending upon how much progressed it is. But adults can have it until the sutures become ossified. Now, question?
So, say a patient has severe head trauma and they're bleeding from their ears, CHF, weak outlet, correct?
Yeah.
What would result in this pathway then? Like what would happen?
When you lose CSF, okay, then the pressure is going to be reduced within the brain. So what most likely you're going to cauterize, they're going to try to close the vessels so that they're not leaking anymore. Sometimes they might have to go in through the skull, in the portion, and be able to cauterize it. But the bottom line is that you produce it nonstop. You're constantly producing CSF. So there is a circulation pattern. The CSF that's produced in the ventricular system eventually goes up to the superior sagittal sinus, and it will actually empty into the venous return from the head, and then it goes into your bloodstream. So you do have a circulation, and the fluid then just goes into that. So yeah, you've got a lot of severe things going on when you have that extent of head trauma. Okay, all right, so that's the ventricular system. Now, let's look at some of the features associated with the cerebrum. This is an actual head. I don't know how they do this, when they cut everything up, but this is actually the cerebrum within the skull. You can see its orientation. You can see the two cerebral hemispheres. There will be in the center here a longitudinal fissure that separates the two cerebral hemispheres from each other. And you keep, there is a membrane that goes down in between into the longitudinal fissure to make certain that the two halves are separated. Again, it's for protection, so when you have trauma, the brain will obviously respond to that trauma, and so then the ventricular system is going to reduce some of the spread of that particular head trauma, and then it's going to come over and it's going to hit kind of a wall, so that the wall, this meninge right here, is going to then prevent that trauma from going over to the other side. Okay, so you have some safeguards within the brain that help to maintain and protect it as much as possible. So we've got fluid in the form of CSF. And CSF, by the way, just like blood, is sterile. So there should never be any white blood cells in there. White blood cells tell you there's an infection. So when you have something like that, then that tells you you might have something like a meningitis or an encephalitis or some other kind of condition that's going to result in an infection. But that should be sterile, along with your blood. That should also be sterile. Okay. So you see then the partition, okay, and you see all these convolutions. Now, you can see that there are a number of folds that are associated with the cerebrum. In fetal alcohol syndrome, the most severe case I've ever seen is that when the mother is drinking alcohol during embryogenesis and fetal development, it's going to affect then the central nervous system. And you can see how numerous these folds are. I've seen one brain that there were only 3 folds. Okay, so that reduces the surface area for your cell bodies, your neurons, etc. So that baby didn't survive. It was just too much. I mean, there wasn't enough to keep everything functioning there. So the convolutions are important because they serve as the area in which we're gonna find our cell bodies, okay? So here you can see this tan colored, all of that is white matter. So what's white matter? Myelinated axons, right? Predominantly, that's what it is, myelinated axons. And then what we find, and sometimes we might find, and then, let me continue on here. So this outer part is where it's gray matter. Gray matter is where I'm gonna have my cell bodies. So the axons coming from the cell bodies will go into the white matter. And the white matter then is gonna allow for nerve impulse transmission, because isn't that where the action potentials are, along the axonal lemma? And if it's myelinated at the no neuron day, you're gonna have exposed axonal lemma, where you're gonna have then the action potential, okay? And then you're gonna have then communication. This is white matter. This large structure allows for communication between the two halves of the cerebral hemispheres. Okay? And you can see then all this white matter versus gray matter. Okay? Again, gray matter is the location in which you're going to find the cytons of my neurons, okay, of my neurons within the CNS. Okay? Now the corpus callosum, let's look at it. This is what we were looking at here. Okay? It's going to connect the two cerebral hemispheres and allow for my interneurons, the axons of my interneurons going in there to then allow for that kind of communication to coordinate the activities of both cerebral hemispheres. We don't want just one side functioning and the other one not. When we have pupils that dilate, they should be dilating simultaneously. That ensures that we've got some, there's functionality. One of the things that you check for when you have head trauma is you look for the pupillary responses or the pupillary reflexes. If you find that one is enlarged but the other one is non-responsive, okay, well there's damage going on in there, okay, because that means then you've got some kind of a interference in nerve impulse transmission, because they should be simultaneous. Reflexes are going to be simultaneous onto the one side, or onto both sides. Okay, now this is what the corpus callosum is going to look like on a mid-sagittal cut. So again, this is relatively large. It is composed of myelinated axons, and that then allows for the communication between the two cerebral hemispheres. This is what it's going to look like on a coronal section. Okay, this is depending upon where you've made the cut. If you make the cut here, you're going to see something like that. If you go back further, back in this area here, you're going to see something like that. This represents then my ventricle. There, you can tell that. So here I have everything out here that's dark shaded is gray matter. And we know it's found in gray matter. So what do we call this gray matter here? These are islands of gray matter within the CNS. What do we call them? Generically. Nucleus. Okay, so this is a nucleus. We're going to learn the names of these various nuclei that are present here. Sometimes they're referred to as basal ganglia, okay? So it's kind of confusing because you go, oh, there's spinal root ganglia or dorsal root ganglia, and then we've got some basal ganglia. What's the difference? Well, the basal ganglia are within the CNS. The dorsal root ganglia is in the PNS, right? So the cytons of the unipolar neurons are in the dorsal root ganglia in the PNS. Okay. The cytons of the unipolar neurons. That means that's sensory information coming in to the spinal cord. Those cytons are located in the dorsal root ganglia. That's part of the PNS.
So, unipolar, PNS,
information coming from, like, continuing corpuscles or your miters. It's coming in. These are my dendrites. There are nerve receptors there. There are cells that pick up the information. It then carries it. It's a sensory impulse, so it's an afferent neuron going toward the spinal cord. The cell body of that neuron is in the dorsal root ganglion, and then it brings the axon into the spinal cord. Once it goes into the spinal cord, I'm going to have a synapse with an interneuron in the spinal cord. So unipolar are sensory neurons, aka afferent neurons. The cell body is found in the dorsal root ganglion. The multipolar neurons, they are found in the gray matter within the CNS, because the information to go to my motor response is going to originate from within the CNS. Okay? And then the bipolar is also, it's sensory, right? And it's only found in the retina and the olfactory mucosa is part of the olfaction. And so then you're going to have a dendrite coming in. The cell body is going to be in my nervous system, and then I'm going to have an axon going away. Okay. So there's my corpus callosum. Now this corpus callosum is going to it's going to allow for association between the two halves, so they are association fibers that are found there. I'll go over that. Okay, so here we have another representation. This one here has been somewhat stained, okay? So you can see the white matter versus the gray matter. The spaces that are in here are going to be my ventricles within the CNS, okay? And you can then see these islands of gray matter, which are then the nuclei. This portion here is my pons. This is kind of like making a cut here, and then I'm looking back to see the pons, and then here's my medulla. That constriction right there is going to then lead down the spinal cord through the foramen. Didn't have enough caffeine. Okay. All right. So some information regarding the cerebrum. Okay. There are two halves. Again, we have to make an association between the two so we can coordinate the activities between the left side and the right side. It accounts for about 83% of the brain mass, so it's the largest portion of the brain itself. You can see that these little folds, I call them collectively the hills and the valleys. They're called convolutions. That's the generic term for the hills and the valleys. But I can break it down to the hills, which are called the gyri, that's plural. A single gyrus, okay, is the hill. And then the sulcus is the one that goes down, or sulci is plural. That's all this is showing you. Okay, so we have the convolutions that are then going to be a result of these gyri and the sulci. Then we have these long, deep openings. Those are called fissures. And we've got several fissures that help to separate various parts of the entire brain. So we've got the longitudinal fissure, which goes down here, and you can see that it's going to separate the two cerebral hemispheres. That longitudinal fissure goes all the way down until it hits the corpus callosum. Okay, so it goes down to the corpus callosum. And then you have a transverse cerebral fissure because there is a space between the cerebrum and the cerebellum. So I've got then meninges that go in there, and that opening, or that fissure, is called a transverse cerebral fissure. So separating the cerebrum from the cerebellum is this fissure. You'll be able to see it in the models so that you're able to identify it for a lab test. Name the opening, okay? And you'll see it going all the way across, and you can see the separation between the cerebrum and the cerebellum. Okay, so here are, just diagrammatically, an enhancement of the gyri. Those are the upward hills, and then the sulci, which go down, okay? And collectively, they're called the convolutions of the brain. You can see on this brain, you can see then this longitudinal fissure, which extends from the frontal lobe all the way back to the occipital lobe. By the way, the cerebrum has 5 different lobes associated with it, and the name of the lobes refer to the bone under which it is found. So the frontal lobes are found in, or deep to my frontal bone. Okay. My parietal lobes are deep to my parietal bone. The temporal lobes are deep to the temporal bone. Okay, so it's pretty easy to identify. Now you can see that this one here has been stripped, this side has been stripped of all the blood vessels and all the protective membrane, the one that actually sits right on top of the brain itself. And this one here, you can see that you've got a number of blood vessels, and these blood vessels are pretty superficial, and these are major arteries. So underneath my temporal bone, did you see how thin that bone was? We had a temporal, if you weren't able to see it, I'll take out the temporal bone. You can literally hold it up and you can see light coming through it. Okay, that's how thin it is. What protects that temporal bone is your temporalis muscle. Okay, but deep to my temporal bone, just right on top of it, is called my middle cerebral artery. And if that's severed, you can't get in there fast enough, and the person is going to die of hemorrhaging. It's a major blood vessel just underneath that temporal bone. So it was godsend that we have these motorcycle helmet laws to protect then some of the trauma that can be, induced on the brain as a result of, whatever collision you might have with your motorcycle. But you can see then, highly vascularized. Okay? That's why the brain requires, it's about 6% of all the glucose at any given time from your blood. It requires a lot of glucose in order to be able to function. Okay, so let's see. Okay, so if you look at this is a lateral view of my cerebrum. And again, on the models that we have of the cerebrum, some of them are color-coded so that you can tell the various lobes from one another. And you can see then a partition. So these partitions help you to identify the various lobes. You can only see 4 of the 5 lobes superficially. So if I'm looking at this from an anterior view over here, this is my frontal lobe. Then you can see the parietal lobe, the occipital lobe, and the temporal lobe. And you can see that those lobes are going to be separated by way of relatively deep fissures, okay? Or actually sulcus. So the central sulcus is this right here. Okay, so it separates the frontal lobe from the parietal lobe. So on the brain model, you're going to have a piece of tape and it will say, name that opening. So that would then be the central sulcus. And you'll notice that on that central sulcus, which separates that frontal from parietal, there's a gyrus just in front of it and a gyrus just in the back of it. And you're going to have to know what those gyri are called. Okay, so just want to kind of give you a preview about that. This is my lateral sulcus. The lateral sulcus is going to help to separate the temporal lobe from the other adjoining lobes. Then the parietal occipital sulcus is going to be this one here, and it then separates the occipital and parietal lobes. Okay? And this one you can't see as prominently. This one you definitely can tell. This one you can definitely tell. So expect to be asked that on the lab test. Name that opening. Okay. Okay. So 5 lobes are listed for you. Got that on the handout for you. So the 5 lobes, and some of these, they're all paired. Right? Okay. So the frontal lobe, parietal, temporal, occipital. The only one we can't see superficially, and there is some disagreement as to whether or not people want to call it a lobe, but we're going to call it a lobe. And that would then be the insula. Now the insula, let me go back a moment. This here is going to be my lateral sulcus. And if I take and, let's say I put my fingers in there and split it, okay? Then if I look deep to that, that's where I'm going to find my insula. So the insula is deep to the lateral sulcus. Cannot be observed from a superficial view. So you can see again, this is a cut through here so you can actually see it. So the reason they could tell that that is a fifth lobe is because it's lined with gray matter. Just as we see on the surface, where we have the gray matter here, okay, on the cortex, you're gonna see that going in as you approach then that insula. So this pretty much identifies, using the color coding on this cerebrum, the various lobes from a superficial view, and then the insula is deep here. You can see the central sulcus. On our models, they're pretty well pronounced, so you can tell what they are. But notice that just in front of my central sulcus is called my precentral gyrus. Now that makes sense. It's a hill, so it's a gyrus. Pre means just in front of. Precentral gyrus, okay? Just behind, I'm gonna find then, okay, just behind, I'm gonna find my postcentral gyrus. Everything that you see here outlined in this red area, part of the frontal lobe, but it's also motor information. So all of my motor function is going to come from the area anterior to my central sulcus. Everything back here is sensory. Okay? Has to do with senses. So my central gyrus, okay, is my primary motor gyrus. The postcentral gyrus is my primary sensory gyrus. Okay. Now the way that I have to remember things like this is that most cars have the motor in the front. So on the anterior side, it's going to be precentral gyrus is motor function. It's in the front. Then sensory is in the back. So that means that all the sensory information is going to come up to that the postcentral gyrus, be interpreted, goes over then to the precentral gyrus and goes back down. So the information goes into the spinal cord, goes on up, makes its way to the thalamus, ultimately makes its way to this, and then we're going to have, this portion here tells me where's my sensory information coming from. How do I know it's my toe and not my thumb? Well, it's in this postcentral gyrus that tells where that information is originating from. And then when I have a response, if I have my toe, if I hit it into a door or whatever like that, I'm going to move my foot back. Well, the information's got to go to the precentral down so I can move my foot back. Okay, so my motor comes from my precentral gyrus. And we'll talk about the function of that when we get to the spinal cord. Okay, so this then shows, going through the lateral sulcus, the insula. So it's like pulling it apart, and you can actually see that. When we used to, this is a really neat dissection on a sheep brain, but again, we don't do side dissections anymore. But it's a real easy one. Cut through that lateral sulcus, pull it back, and you can see the insula very easily. Now you can go to some stores, they actually have like sheep brains. Some people like to eat sheep brains. I don't know who. Organ food, organ meats are just really, they're kind of got a weird texture like liver. I don't know. Anything that's organ has got a weird odor and a weird texture. But some people enjoy it. Okay, so you then can actually see that. So this is where it's actually been cut away, and there's my exposed insula. I see convolutions on inside, okay? And that's then going to be my insula, my fifth lobe. So again, just going through and seeing the various lobes, we're going to go through and give a little bit of function associated with them. Okay, so this is a major controlling center of your entire body. So everything that you sense, everything that you respond to, has got to be integrated into that. Okay, into that area to be able to then respond. So let's look at the frontal lobe. This is easy enough, we already know where it's located, okay? But this is where we're gonna originate with, now these are not my slides, remember? Okay, so, the frontal lobe is associated with voluntary motor functions. So, as I'm speaking, the way my tongue moves, the way my jaw moves, the words formed in order to express, that is all under the control of the frontal lobe. Lots of your social behaviors, your critical thinking, your ability to be analytic, that's all associated with the frontal lobe. And a lot of research now shows that the frontal lobe is not fully mature until about 26 years of age. So it's no wonder then that sometimes the critical thinking ability of teenagers is really not there. You just kind of have to say, well, you know what, not fully developed yet. That's what you have to say, because there's nothing that you can, there's no other way that you can explain some of the bizarreness that goes on in terms of social behaviors, in terms of how to critically think through anything. It's true, isn't it? Yeah. So I just have to say, if they're younger than 26, I just go, it's frontal lobe, it's not there yet. So when my grandson does strange things, I go, it's frontal lobe, it's just not there yet. He's 21, okay, personality, everything that's associated with who you are is associated with that frontal lobe. The manic depressive behaviors that you have are associated in your, the violent behaviors are associated with frontal lobe. So way back when, not way back, like we're talking in the 40s and 50s, when mental patients had outbursts of violent behavior, they'd do a lobotomy on them. You would actually go in there and you're gonna go right through the orbit of the eye, get up into the corpus callosum, into the frontal lobe, and disrupt it. Then there's no communication, and they become silent pretty much because there's no behavior. The behavior has been pretty much eliminated. Okay, and that was the way that they would do it. And how do you kind of knock them out to do that? Well, you do electroconvulsive therapy. You shock them. Really, you just shock them, and then they go out, and then you do the lobotomy. I mean, it just, you think about these things and you go, I cannot believe that was common practice. Okay? I'm going to show you a slide, I think, that of this person who actually had kind of a lobotomy of vehicle, RV, basically. And he would go around and do lobotomies. I mean, it's just ridiculous, but that's the only way that they thought you could control behaviors. They didn't understand neurotransmitters and the chemical imbalances that you can use medication to help eradicate that kind of behavior, or at least minimize it, right? So that was the way that they would do it. And this shows how they would do it. It's pretty vicious. Okay, go right in there to do it. And again, these are not my slides. Okay, but this guy here, the lobotomobile, he did as many as 4,000 patients, 25 patients a day. I mean, can you imagine doing that and having the apparatus there, just kind of did the electroconvulsive therapy? Because really, I'm going to talk about brain mapping. When they did brain mapping to figure out what part of the brain controls this part, they literally took off of mentally ill patients. They were the guinea pigs. They'd take off part of the skull and then put an electrode in there and see what would happen. And if the leg kicked up, you go, okay, that area there is controlled by, the leg is controlled by that portion of the brain. That's literally how they did brain mapping way back then. Yeah, questions?
What year?
We're talking in the 40s. Not that long ago. Okay, so we've got special areas that are important. So in your lab manual, you have a list, a diagram labeled with various regions that you need to be able to identify. So that would then be identifying a place such as Broca's area. Broca's area is the area associated with the frontal lobe that's important in making speech. All the muscles that are associated with speech formation are going to be here. Then back here, Wernicke's area. Wernicke's area is how you construct your sentences when you're speaking. So if you have a stroke here, you may not be able to speak. Okay, which means then you go, oh, Broca's area has been pretty much, and it's only on one side, it's on the left side. Okay, so what you have then is the motor region is not responsive. You've got blood from a hemorrhage, okay, from a stroke, which is then going to interfere with nerve impulse transmission. So we're not getting any information going there. This part here is how you construct your sentences. So if you've got a stroke back here, instead of when I'm talking to you and I'm constructing a sentence, I can have what is called word salad, where the words are not in the right sequence. They just are kind of like, they sound like, what goes on here? The muscles are moving, that motor information is going, but the information sensory to get to that is not connected. Could be, when you have word salad, the words are just not in sequence at all. Then you think to yourself, well, damage perhaps associated with stroke. Okay, could be a multitude of other things, but primarily stroke. So this is then my association area for verbalization, where I'm creating then my sentence sequence. Okay, all right, and you can see then, these are all sensory back here. Okay, back here, these are all going to be my motor. Now I do have some information relays in here that have to do with memory. So when I have memory formation, it initiates in my frontal lobe, but it has to go into and through my diencephalon. It's got to go through my thalamus. So the way that I can get that information put into the files is that you have to have sleep. So there's a correlation between sleeping and effectiveness in working skills, whether it be in classes. In order to lay down memory, you have to have 6 hours of sleep. How many of you have 6 hours of sleep? I know as a college student, I'm lucky if I've got 3 to 4. You know, keep myself wired all the time. I was taking 21 quarter units. It's just like, I've got to be up and studying all the time. And never really understood because nobody told me. Okay, I wasn't into psychology. I was pre-med. I didn't care about psychology. I'm into the STEM areas, okay? So, all those psychologies, I don't want to argue about that. But bottom line is that, I didn't quite understand the importance and significance of sleep. So I figured I'm up, I can go ahead and do that, and you get to a certain point in time, like about 1:30 in the morning, you can stay up until 6 or 7. It doesn't even matter at that point in time. I can't even sleep after that. So that's what I would do. I would reach a certain point, and then I just need to get to that point, and then I could study as long as I needed to study. But did I have any memory? No. I mean, how many times did I, procrastinate and cram study for an exam? I don't know if you do or not, but I sure did a lot. And try to, I don't need to sleep. I've got 3 days to study the genetics of English. And that means 24/7, 24/3, okay? So I'm going to just study. I go into the test, and I'm sitting there, and I'm going, what the hell is this sentence even saying to me? I went in one time, and I'm going, name, and I'm going,
Oh.
okay. And I go, why am I, I'm going, what? And then reading the sentences made no sense. You know how when you just cram for a test, it doesn't go anywhere. You feel like you're doing your work, but it doesn't go into a memory bank. And you don't realize it until you take the test and you go, dang, what is that? I don't even understand what that word is, what that is even saying. We all go through it. We're all, even me. Okay, we all went through this, and then you learn when you go to graduate school, you know what, I don't need to cram this stuff. I need to study this stuff and then get some sleep so I can lay down. So I can go put it into the right files. I need to do that during sleep. Babies sleep because that's when growth hormone is released. People who don't sleep gain weight. So if there's a correlation between weight and lack of sleep. So there's a lot that happens during sleep that we often forget about because we're just too busy to really take the time to sleep. But if you just plan it, it kind of helps, but I do. Okay, so your memory has got to go through not only the thalamus, but ultimately through the frontal lobe, because that's where you do your critical thinking. And that, once you identify it and you understand it, then it goes into the file. But if you don't understand it, it's like, what file do I put it in? I don't even know what the subject is. So, you can see all of that's associated with solving complex multitask problems. Okay, all of that's associated with the frontal lobe. Okay, you had a question? Okay, got it. Okay, then over here in the back, in the posterior portion, in the posterior to the central sulcus, you can see that this is where you have somatic sensation. You have then taste, which is then still in that temporal lobe. That is still going to be then your sensory lobe. Your hearing is in your temporal lobe. So you can see all of your sensory function is back here. You see from your occipital lobe. Okay. Yes, the image comes through the eyes and then goes to the retina, but then the interpretation or the image goes to the back on the occipital lobe in order for you to actually see. Okay, so everything back here is sensory. So if you just use that guideline to help you when you're studying and identifying, it will definitely help you to separate, because if it's anterior in the frontal lobe, it's going to be motor. If it's going to be then or thinking, critical personality, all of that is over here. And then here is actual interpretation of your, by your senses. Now, everything that goes to that posterior portion in the post-central gyrus and posterior to that passes through the thalamus. The thalamus gives you a memory. So when you smell something, like a rose, when you smell that rose, it passes through that thalamus, and the thalamus then interprets it as an odor, and then files it as a rose. Peppermint, chocolate, all those different scents are passed through the thalamus and then make their way into this portion of the brain. Now, in addition, there are certain memories that are associated with scents. So when you smell turkey, okay, it reminds you of either Christmas or Thanksgiving, right? Holiday season. Okay, and then that might just simply bring up a lot of memories associated with that. Okay. So there is memory associated with the sensory input, is what I'm saying. So we'll talk about how that sensory, or that interpretation of that sensory input, where it's going to be located. Okay, so you're going to be asked to identify that on all of this. What's labeled in your lab number 20 on a cerebrum. Okay, the parietal lobe. So we know that the parietal lobe is going to be posterior to the central sulcus and just deep to the parietal bones. And it's going to contain information that is somatosensory. Touch, pressure, vibration. So when you're sitting, you have a certain posture because you have muscle spindles that keep you in a position. That's part of somatosensory. Body sensory. So when you cross your legs, again, you're going to have tension on the muscle, and that's somatosensory. Okay, and that's associated with then the parietal lobe. 2-point discrimination. You know, we used to do way back when I first started teaching here, we used to do this 2-point discrimination where you can actually use kind of like a compass. I think that's what that is. Is that one? Is that a compass or what's that? Oh, protractor. Protractor. So that what you can do is you can actually measure the distance between the sensory receptors by using the protractor and then just opening up. So if you were to put it on your tongue, the sensory receptors are very close together, so you can very easily see that, you know, distances between those neurons are very, very close. But if you're going to the back, on your back, skin on your shoulder, the sensory receptors are further apart, okay? We call that two-point discrimination. That is, you can tell that there are two pointed parts of that protractor that are on the skin, okay? Two-point discrimination, okay? So what I was saying about muscle spindles when you're sitting there with posture, those are proprioceptors. They're able to tell you that you're in the sitting position versus a standing position versus a prone position, okay, or a supine position. So that's all found in the parietal lobe. Now this is what I call the brain map, okay? And this then represents the motor, this is my pre-central gyrus, and I have on both sides, left side and right side, information. Now they're gonna crisscross, so my right side of the brain interprets the left side, and the left side interprets the right side of the body, okay? So you can see then that when they did the brain mapping, and they're able to put in the electric probe, they could actually see which parts of the body were going to respond to the electrode, okay? And that then became this homunculus model or brain mapping. This represents going in here, this is my lateral sulcus going to my insula. Okay, so this is a coronal cut. So you can see then going here, this gray matter right there is gonna be part of my insula. All right, so you can see then the portion of the body and location of the body in which the sensory or the motor receptors are going to lead. So if I want to move my foot, I'm going to have to then activate down here. Okay, let's see, my foot down here in order to respond to a stimulus. Okay, this is where the sensory input comes, and that's found in my post-central gyrus. So they were able to then identify from which area of the body is the sensory information originating. And you can see then, notice how much of the surface area of the gray matter is associated with, for example, the appendages, the face, okay, then internal organs, etc. Okay, so anyway, this then is my brain mapping. Now, you're going to get a lot more information about this in the 50B because you're going to actually analyze the pathway in which information is going to be relayed. And you can see right here, this kind of shows the precentral gyrus, the postcentral gyrus. So the question on the lecture test would be, which of the following areas of the brain would you expect to see then? The origination of the motor response. That's coming then from my precentral gyrus. That's where it's going to originate. Where's my endpoint for my sensory information? It's going to be at my postcentral gyrus. Okay. Temporal lobe. Okay. It's again deep to the temporal bone, and its primary function. Just think about where it's located. So I'm trying to make sense of this because if you're just memorizing, oh my gosh, there's too many factoids in here. So if it's close to my ears, oh, that makes sense. You're going to have auditory information coming in here. Okay, so that makes very good sense. Incoming auditory and also olfactory information. Now the olfactory, the sense of smell, actually is going to go through that temporal lobe, but it will make its way back to the thalamus as well. So the temporal lobe, again, this is just a kind of a summarization of what that area would be. Occipital lobe. Okay, so occipital lobe is deep to the occipital bone. Like I said, the names of the lobes reflect the bone under which they are found. So So it makes it easier for us to identify. Just orient yourself when you're taking a test, which one's anterior, which one's posterior, right? Before you start identifying the lobes. But you can see the occipital lobe is where we're gonna have analysis of the visual stimulus. So information comes in from the receptors, makes its way back then to the occipital lobe, where then I'm going to be able to interpret what I've been seeing, and it also is going to store my visual memories. So it's going to be back in my occipital lobe. Last one I'm going to do in here right now is my insula. And the insula is, again, deep to the lateral sulcus, and it's used for the interpretation of taste. So I use that one to identify, when you're blindfolded and you put something on your tongue and you're trying to figure out what that is. Okay, that interpretation is accomplished by way of the insula. And you can see here the pathway in which the information is passing. So this is going to be the predominance of your 50B. You got to know where these various pathways are originating from. The taste. We always start with the sensory receptor. You got to have something that's interpreting that stimulus. So it's got to start on a cell. Pacinian corpuscle. What kind of information is being interpreted there? Deep pressure. If I said Meissner's, fine touch. Okay. If I'm looking at my retina, I'm going to have the retina has cells, photoreceptors, that are going to interpret then what you're viewing, and then it's going to come up into my thalamus. That's this island of gray matter here, and I'm going to have a synapse, and then it's going to go over to, or in this case, for the taste, it's going to go over into the insula. Okay, so that's how the information is going to go in terms of how the brain is going to interpret this. So if we did a neuroimaging, you can see which parts of the brain become very active when you're looking at smelling, when you're stimulating a sensory receptor. So you've got then these areas. It kind of reaffirms what we've already identified as terms of areas on the brain with respect to the function. Okay, so tonight your job is to at least look at the superficial brain. Get an interpretation, be able to then identify using the labels, answering questions, okay? And then